Here's a quick review about meiosis and genetic variation. So the question is, what would happen if reproductive cells, your egg and your sperm, were diploid? And here's a little picture. We got some peas that are diploid. Their diploid number is 14. And if they made cells, gametes that were diploid, you'd end up with, when the sperm and the egg come together, with 28. That is too many. Too many chromosomes. Have lots of issues. In fact, this probably would not be an individual. So when we do meiosis, it is extremely important that, again, just going back to mitosis, that mitosis makes cells with a full set, with a diploid number. Those are your body cells. But when you make your gametes, right, you need to cut those chromosome numbers in half in order to get a full set when the egg and the sperm come together. And that's what meiosis is all about. So if we look at our picture again in these pea plants, if the sperm and the egg are haploid, then we come together and we make a diploid individual. So that's what we want. That's what meiosis is all about, making the gametes that are half the amount of chromosomes. So the stages just look very similar to mitosis. It's really just mitosis times two. We start with prophase, right? Chromosomes become visible, nuclear membrane breaks down, and in metaphase, this is a small difference. In metaphase, the chromosome pairs line up in the middle. And in metaphase, in mitosis, you don't have pairs, you just have single chromosomes lining up in the middle. But here we have pairs that line up. In anaphase, the pairs separate. And in telophase, cytoplasm starts to divide to make two cells. But we've not, again, going back to this picture, we have not reduced our chromosome number by half, so we still have to do that. So in prophase 2, similar things. We have certain things like centrioles and the spindle fibers reform, and so we're just getting ready to divide. Now in metaphase 2, now we have the individual chromosomes line up in the middle. In anaphase 2, those individual chromosomes separate, and in telophase 2, you can see that cytokinesis begins and the nucleus will begin to reform. So what we end with, again, at the end we have 4 cells, not 2, but 4 cells that are haploid, that have half the amount of genetic information that we started with. Now that's some good stuff. Now that if these are sperm or these are egg, that when those sperm and egg meet, we're going to have a diploid individual. So that is perfect. Now we're all set. So if we look at the differences between mitosis and meiosis, we're still going to start with one diploid cell, full set of chromosomes. But in mitosis, we end with 2 cells. Meiosis, we end with 4. The number of chromosomes is where it's going to be different, because in mitosis, we're making body cells. We're making cells that are identical to one another and that are diploid, that have a full set. In meiosis, we're making cells that are haploid, that are half. And these actually are very unique cells. Each cell is unique, is different, right? Based on something called crossing over, which makes all our cells different. And we'll talk about that in just a minute. But this is a really good comparison between mitosis and meiosis. Body cells for mitosis, gametes for meiosis. Mitosis makes 2 cells that are diploid and identical at the end. Meiosis makes 4 cells that are haploid and very unique or very different. So if we look at why this happens, why we get in meiosis cells that are so different, and it really has to do with something called crossing over. Crossing over, it happens in prophase 1, where the genetic information just simply crosses over, exchanges itself. And you can see that whenever you exchange DNA, that's going to create some different combinations of traits. Here's another picture of it where the genetic information just does a little dance and crosses over. Different combinations of traits makes us all different. If this little phase did not happen, then we would all be looking at everyone being the same. It's very important that this process happens. And you can see here more of this is what happens when the chromosomes get separated. Right? Maybe one go around, we have the chromosomes being made like this, where we have these different combinations of traits in all 4 cells. Maybe a next go around, they split a little differently. So we have a different combination. So in meiosis, every new cell that we make is going to be different from the starting point. And that's important to our species because we need genetic diversity in our species. So that concludes the review for meiosis.